We are sorry about the delay is because of electricity and we had to change the systems and stuff. So that's why this happened in that regard. But nevertheless, we are here. Let's start without wasting time. But before we start our discussion, I want us to go through these terminologies. Just very fast, in 1-2 minutes, we go through the terminologies. At least we know that we are covered with that. Yeah. All right. Let's go through very fast. They're saying that number one, so let's use 2 minutes so that we are done with this. The allele that does not influence the phenotype when found in heterozygous state. So this allele, it does not influence. So it means that it is a recessive allele. So the position of a gene on a chromosome, we call it a locus. Then you're saying that the physical and the function expression of a gene, how do we call the physical and the function expression of a gene? We call it phenotype. The chromosome that are not responsible for sex determination, we call it autosomes. Remember, we have the gonosomes and the autosomes. So it means that from chromosome number 1 to chromosome number, I'm talking about the pairs 22. So we call them autosomes. Then they're saying the process of finding a desirable gene. You find the desirable gene, isolate that gene, and then move it to the cell of another organism. So we call this one genetic engineering, or because you are manipulating these genes. Yes, or modification, genetic modification. You are modifying the genetics of another organism. Then we go to the next part. As we said that, let's first go through the terminologies very fast, and then we go to the content we are interested in more, so that we don't lose these 10 marks. The two parts of the chromosomes held together by the centromere. Two parts of the chromosomes held together by the centromere, so they are looking for these parts. So we call them chromatids. Section of DNA molecule that code for a specific characteristic, a section of DNA that code for a specific characteristic. We call it a what? A gene. Then you're saying that the production of genetically identical copy of an organism using biotechnology. So you produce the same individuals which are identical. So we call it cloning. So we are saying that deliberate breeding of organisms for desirable characteristic selected by human. Can talk about artificial selection, because you do that, you select artificially, you select by the help of the manpower. Like what you want is what you make the organism to continue. But if it was done by natural, then it would have been natural selection. So you guys, if you have any question, you let me know. Yes. So I'm going through this as you also do that, so that you know that when I say something, you get it right. Né? Yes. So make sure that before I say the answer, you also do the same. Hey guys, don't forget that you are focusing for a distinction. Then you're saying that the deliberate breeding of organs, so we are done with that. The explanation of the species experiencing long periods of without physical change followed by short periods of change. Now, I think in your mind, you have the answer. So when I say it, it must be, we are testing ourselves now. So it means that it looks like this. Long periods of no change, and then short periods of what? It must go like this, the ladders. We call it punctuated equilibrium. So if they bring two graphs and one is like that and one is like the ladder, then you just know that steps, you know the steps. Yes, you just know that that is called punctuated equilibrium. Then the phase of meiosis, during which homologous chromosomes separate and start moving towards the opposite poles. Homologous chromosomes are being separated and then they move to opposite poles. So they're looking at the next phase after this, meaning that they have to separate, they move to the opposite poles. So it is anaphase 1 because they're saying homologous. Don't just say anaphase, it must be anaphase 1. The defect in the cell division that lead to Down syndrome. So how do we call that? defect in the cell that leads to Down syndrome, we call it non-disjunction. The structure that is made up of two chromatids joined together by the centromere. Structure that make up two chromatids joined by the centromere. So it means that structure, yes, structure made up of two chromatids. These are two chromatids, and then joined by the centromere. How do you call this? It is a chromosome. Then they're saying that an explanation of something or for something that has been observed in nature and which can be supported. When they say supported by facts, laws, tested hypothesis, then automatically it can be hypothesis. It is a theory. Then they're saying that, remember when they're asking you this, it is either theory or a hypothesis. So if they say tested hypothesis, definitely the answer can be hypothesis. The answer is the theory. We go to the next question. Guys, if there is anything related to the sound and the sound is not clear, let us know. Maybe there is echo, maybe you don't hear me properly, something like that. Or I'm too fast, you let me know. Yeah. But because you wanted to finish this in 2 minutes, at least, yeah, the similar structure of different organisms that suggest they have a common ancestor. Similar structures. Whenever you say similar, it is homo. Starts with the word homo. So structures that structures means logos. When you say homo, similar, and then structures is logos. When you say logos means structure. So it is homologous structures. Then they're saying that a complete set of chromosome in a cell. How do you call that complete set of chromosome? Is a genome. So you guys, you confused, sorry, did I say genotype? You confuse three things: genome, karyotype, and a genotype. Genotype is a description of organism in terms of genetic composition or in terms of gene. And then when you talk about the complete set of the chromosome in a cell is a genome. And then when you talk about the arrangement, it must have the word arrangement. The arrangement of chromosomes, we talk about size and talk about the structure. That's when you talk about the karyotype. I think I'm clear there. A barcode pattern formed from DNA is DNA profile. Then the bond formed between two amino acids. Guys, this bond is called the peptide bond. Please take note of this. We don't have what you call polypeptide bond. It's peptide bond. If there are many bonds, we call them peptide bonds. We only add the word polypeptide if we are talking about a chain. So if you don't add the word chain, then you can talk about poly. So it's polypeptide chain, peptide bond. Is it clear? Yes, please, if you add the word polypeptide bond, is wrong. You will get a zero with big eyes, and those eyes are looking at you. So make sure that you don't say polypeptide bond. It's peptide bond. Okay, we continue. The representation of the number, shape, here is the answer. Representation of the number, shape, and arrangement of chromosomes in the nucleus of a somatic cell. So is what you call the karyotype. Opening in the nucleus, the nuclear membrane that allow messenger RNA to leave the nucleus, nuclear pore. So we have got it. I'm not writing the words because we want to finish up this so that we go to the major section. They're saying that a tentative explanation of a phenomena that can be tested. A tentative explanation of a phenomena that can be tested. You see now, it's a tentative. So it becomes a what? A hypothesis. Yes. Distribution of species in different parts of the world. Different parts of the world is geography. Different parts of the world, but distribution of species, so it must be linked to bio. So it becomes biogeography. Then they're saying that chromosomes that carry same set of genes. Chromosomes that carry same set of genes. How do you call those chromosomes? Homologous chromosomes, meaning that when they look at it, they look like that. They're saying two or more alternative form of a gene at the same locus. Two or more alternative form of a gene at the same locus. What does it mean? Two or more alternative form of a gene. It means that either this or this, for example, or capital R, small r, something like that, we call them alleles. So they call them what? Alleles. The structure responsible for pulling the chromosome to opposite poles of animal cell. So opposite poles. You remember when you are like this, how do you call that structure which pulls the chromosomes to opposite poles? So we call it spindle fibers are the one which pull. Yes. Then they're saying that a phase in the cell cycle that occur before cell division, interphase. Cell cycle, a phase in the cell cycle. Yes, it's not, they're not saying the phase in meiosis. They're saying cell cycle. So it means that you talk about interphase. So now, let me ask you a question. Who has got all of them? 26 out of 26. I hope you are getting 26 out of 26. Yes. I'm seeing people are getting answers correct here. Thank you very much. Thank you very much. Sir, the sound is making it hard to concentrate. Let me see. I don't know the sound. Which sound? Is there any other sound behind it or something like that? I don't know. Let me know if there is something like that. All right, let's continue, guys. Let me know if there is any awkward sound behind my my what? My speakers or something like that. Yes, so that I can try to work upon it. All right. Let's continue, guys. They're saying that let me see how many people got it. Or let me check how many people got all of them. Someone is saying that it's gone now. It's gone now. Oh, thank you. All right. Let's continue. They're saying that diagrammatic representation showing the possible evolutionary relationship among organisms. Diagrammatic representation, yes, possible evolutionary relationship. Here we're talking about phylogenetic tree. Yes. The type of vision shared by two humans that allow for deep perception. Stereoscopic vision, yes. We call it stereoscopic vision. A genetic cross involving two characteristics. We call it dihybrid cross. They're saying that because two means di. Yes. A genetic disorder characterized by absence of blood clotting factor, hemophilia. Present day distribution of organism. Present day. How do we call about the present day distribution? How do organisms being distributed? So we can call it biodiversity. Yes. We can call it what? Biodiversity. Then they're saying that the nucleic acid that the nucleic acids that carry amino acid 2, the ribosome, transfer RNA. Then they're saying that the natural shape of DNA, double helix. Guys, don't say double helical or helical. We will cross it. It's double helix. It can't be something else. That's it. Double helix. Section of DNA that code for a specific protein. Section of DNA that carry for a specific protein. Okay, let me read again. Section of DNA that carry for a specific protein. How do you call that section of DNA? Check. They're looking at something like this. At this section, how do we call those sections? We call them genes. Yes. And then I'm saying evolution with a long period of no change followed by short periods of rapid change. Don't forget that. It is drawing it ugly. Remember, it is long periods of no change, rapid periods of change. Long periods of no change, rapid periods of change, like that. So, sex chromosome of organisms. How do you call that? Gonosomes. The descent with modification. How do you call descent with the modification? Yes, how do we call descent with the modification? Let me see in the comment below. I want to know number 37. Yes. I want to know this answer. Then in the comment, please put it in the comment. Just write 37 and then you put the answer there. I want to see if you're gonna go get it correct. A group of organisms of the same species in specific habitat. Group of organisms, the same species, yes, but they live in a specific habitat, becomes a population. The family to which humans belong, hominidae, not Homo sapiens, hominidae. Human disorder, human disorder caused by non-disjunction, Down syndrome. The stage of protein synthesis during which messenger RNA is formed. How do you call that stage of protein synthesis during which DNA is messenger RNA is formed from DNA is transcription. Then they are saying that the type of variation in humans with no intermediate, we call it continuous variation. Guys, let me just explain this just a little bit. Whenever they talk about examples of continuous variation, anything which we are born and it keeps on changing, we call it continuous variation. Skin color, size, body size, size of the shoe. Check, when you are born, you are putting on size 1, if not -1. I don't know if there is such, or 0. whatever. But now you are putting on size maybe 47. Big, such a way that is bigger than you. So it means that it keeps on increasing. So it changes. What about your blood group? If you are born blood group A, you cannot change to blood group B. Never. So blood group becomes discontinuous, while the size of the body becomes what? Continuous. The size of the color, continuous. Yes. I'm just emphasizing this because I might not give you examples when you reach there, when you're trying to do highlights. The type of inheritance where dominant allele masks the expression of a recessive allele. Complete dominance, meaning that if it's capital T, small t, this one is completely dominant over this. Therefore, complete dominance. Variety of living life forms that exist on Earth is biodiversity. Let me check if there is any comment. There's someone is saying discontinuous. Down is Darwinism. 37 is saying Darwinism. Thank you. J punctuated equilibrium. I don't see people telling me the answer. A discontinuous. It's only one person who did J. Yes. J, since you have tried to answer, you must remind me after the live. Yes, I'll give you 20 rand airtime. Yeah. However, that's not a correct answer, but you have tried. Yes, I'll give you 20 rand airtime. Yes, but I know how I'm gonna find out that it's you. Don't worry about it. I'll find out. Yes, so 20 rand airtime, Jace, is for yours. Congratulations for that. All right. Let's continue. So the type, if you go back here, you're saying the modification by descent J, the answer is supposed to be let me leave it. Let me leave it. Let me see if I can have some more. Okay, someone is telling me, well, leave it to see that if there are some people who are going to participate. But I'm not saying that I'm going to give you a gift. All right. They're saying that a variety of life forms that exist on Earth, that is called biodiversity. They're saying opening in the skull through which spinal cord enters. We call it foramen magnum. The testable statement that can be rejected or accepted, it is a hypothesis. Total disappearance of species from the Earth, we call it what? We call it extinction. They are saying that a segment of chromosome that code for a particular characteristic, should I leave it also? A segment of chromosome that code for a particular characteristic, we call it a gene. We talked about it. Ability to walk on 2 limbs, we call it bipedalism. The organelles in the cytoplasm, which is a site for protein synthesis, is a ribosome. The name of the bond that formed between 2 amino acids, I explained this. It is peptide bond. Then you are saying that the process by which DNA molecule makes identical copy of itself. Then process by which DNA molecule makes identical copy of itself. Yeah, we call it, should I leave it there? So it's DNA replication. Name the phase, the process where homologous chromosomes fails to separate during meiosis. How do we call that? We call it, they're saying the process is now disjunction, fail to separate during meiosis, but which phase is anaphase, ne? Yes. So we call it what? Now disjunction. Permanent disappearance of species from the earth is extinction. They're saying that testable statement that may be rejected or accepted. I don't know why they repeated it, but is a hypothesis. The type of nucleic acid that carry amino acids, we call it transfer RNA. The selection of breeding, the selection and the breeding of organisms with the desirable characteristics by human. How do we call it? You select the breeding and then desirable characteristics. Since this selection, then therefore we can call it selective breeding. And then they are saying that an allele that is not shown in or expressed in the genotype, in the phenotype when found in heterozygous state. For example, capital T small t, this allele that we are talking about. So you call it a recessive allele. Sudden change in the sequence or the nitrogenous base of the nucleic acid, we call it gene mutation. Explanation of an observation that is supported. That's what is supported by facts, models, and so here they didn't talk about tested hypothesis. But the moment they are supported with this, then it becomes a theory. They are saying that the breeding of organism over many generations in order to achieve desirable phenotype. The breeding, when you breed, yes. So it is basically you are doing artificial selection or artificial selection. Then they're saying that the type of sugar found in messenger RNA, in RNA is ribose sugar. They're saying that the type of evolution with long periods of time when species do not change and short periods of time when there is a rapid change. So we call it that type. We call it punctuated equilibrium. We talked about it. And then he's saying that the mineralized remains of organisms that have lived in the past. How do you call that? We call them fossils. And then the hypothesis which support the migration of ancestor from point of origin. We call it out of Africa hypothesis. Yes. So here is, how do we say this? We say that this is the comparative anatomy. Let me just show you just something small before we go there. Yeah, this is what they are. Another comparative anatomy. Modification by descent. If you look at our distinction material, yeah, it gives you the perfect answer there. Yeah, sometimes they ask you modification by descent, which means comparative anatomy. Sometimes they ask you these structures, whereby you're supposed to tell us that these structures are called homologous structures. So if they ask the process, then it's going to be modification by descent or comparative anatomy. But if they ask you the structures, which is under here, we call it homologous structures. That's why I said, let me know the answers people are giving out. All right, 62 is a ribosome out of Africa hypothesis. Thank you, biotechnology. Lucia, okay, thank you. Yeah, guys, I think now we have got it. Now, let's go to the section whereby what to focus on DNA, what to focus on other topics so that, guys, we don't lose it. We have to look for a distinction today. Yes, the distinction must click. Yes, must click. Yes. I'm going to make it a little bit 2 hours. So we are starting our 2 hours because the previous we are just going through, yeah, and then some technicalities it came up, and then yeah. So now let's start our 2-hour journey story. All right, guys, on DNA, what are you supposed to focus on when you go to DNA? Let me just remove this. Sorry. Yes. Hi. Sorry, guys. Let me just do this. Yeah, we have that. We have that. That. Yeah. Now I think we are done. Let's start now. Guys, on DNA, don't look at that diagram. I'm not talking about the diagram. Guys, on DNA, there are some things which you're going to ask you. And then if you don't know them, you're gone. Yes. Okay, now let's see. Guys, if they ask you, describe the structure of DNA, what do you say? Describe the structure of DNA. Structure of DNA. Guys, when talk about the structure of DNA, look at the characteristics of DNA. So I think that's done. The characteristic of DNA is double helix is made up of two strands. It's made up of the nucleotide. It's made up of nucleotide, yes, which are made up of phosphate, the oxyribose sugar, and nitrogenous bases. You talk about the nitrogenous bases, talk about all of that. Yes. Challenge. Don't go to the paper without knowing DNA replication. Yes, don't go to the paper without knowing translation. Don't go to the paper without knowing transcription. And then describe the description of the structure of DNA. So one will come. Won't tell you which one, but one will come. You'll come. We're going to discuss the paper after. You'll know. And then now, if they ask you a question that give the difference between DNA and RNA, that one is simple. You can talk about everything you know. Make sure that you draw the table. Don't leave it hanging like this. You want a complete table. You mentioning this, you writing like this, and you say DNA, and the other side you say RNA. Guys, put DNA, put RNA, close the table. If you don't know anything, at least you got a mark. Yes. If they ask you to tabulate, draw a table, put the title, close the table, leave. If you don't know anything, yes. If you know something, then write something, yes. I'm done with that. But now the challenge comes in when they ask you that give the difference between DNA nucleotide, nucleotide, and RNA nucleotide. Here, you only look at the nucleotide of DNA, the nucleotide of DNA and the nucleotide of RNA. And there are only two differences in there. DNA has, don't talk about the phosphate because it's the same. DNA has deoxyribose sugar and RNA has ribose sugar. And then DNA has thymine, RNA has uracil. Done. If they ask you about what? About the nucleotide. Is it clear? If they ask you the difference between DNA replication and transcription, you have to know the differences, DNA and transcription. We're going to spend a few minutes here. I'm only going to focus on the things which you fail. Which you fail is what I'm going to focus on. Yes, too much. Then we are saying that DNA, you talk about DNA, in this case, in DNA replication, two strands act as a template. The other side, you talk about one strand transcription, one strand act as a template. Guys, only know at least 3, but we want 2. In most cases, we want 2. We won't ask you more than 2, the differences. Don't go to the paper. If there's a lot, just know few things. Don't on each, at least go have a dose on each, whatever I'm going to talk about. Yes, then you'll be able to make it. Then we are saying that, yes. Then another thing which you go off is about the mutations on DNA. About the mutations on DNA. So when they ask you about the mutation, let me just use my book here so that you understand. Excel in Life Science Distinction Material. Here, let me just, I don't want the structures, okay? I don't want the structures. What I want is this. What I want is at the end here. Yes, at the end, the mutations. Yes. Guys, let's answer this question. That what will happen if a mutation changes triplet number 1? Triplet number 1, it's this, from this, the same thing. From this to this, it means that this has been changed. Is it clear? You changed G to A. So now what you do when they ask you such a question, you first identify what is here. It is a codon. What is given here is DNA. Therefore, I have to convert this into messenger. Remember, we say that, let me do like this. Yeah. Remember, we say that when you talk about code, it means DNA. DNA. When you talk about codon, it means messenger RNA. 3 bases on messenger RNA. When we talk about anti-codon, we mean 3 bases on transfer RNA. So now the question is asking, it means that it's changing from DNA to codon. It means that it's talking about messenger RNA. Is it clear? Yes. So now, what you do is to change this into messenger RNA. So change this into messenger RNA. So when you change into messenger RNA, this is going to be A goes with U, T goes with A, G goes with what? G goes with the C. So I'm going to CUA. I come here, look for C, C, CUA, C. No. AUC. AUC. Where is it? US is triosine. So this is triosine. UCA, isn't it? Then what about this? Now I change it again. Also is U. It's gonna be. It's gonna UAU. What does it mean? UAU is also triosine. So it means that a change from this to this, yes, will lead to this USC, from USC to UAU. That's the first mark. It will, you converting this into codons. You understand? Into codons. Is it clear? Which will code for the same amino acid, triosine. Therefore, there will be no change in the protein. Why? Because it's the same amino acid which is being coded for. Can I repeat it? I said, when they bring such a question, I know when they bring it, and I know it will be there. When they bring such a question, students lose these marks. Change in this, first find out, changing, because here it might be tRNA, and here is codon. So you change into the codon, which is messenger RNA. Is it clear? So change from ATG basically result in this, and the change in this result into this. Is it clear? And these two, they code for the same amino acid. Therefore, it does not code for a different protein. It's like me calling you, let me check. Let me check here. It's like me calling you Lerato, Lo, Lailo, whatever. Ne? Sorry if I pronounced you wrongly. Ne? I can call you Lerato, or I can call you whatever, ne? The second name. I don't want to pronounce it badly. Ne? Yes. So if I call you the other way around, you say yes, sir. When I call you with another name, you also say yes, sir. You understand? So it means that they are coding for the same amino acid. However much the codes are different. Is it clear there? Yes. Then, what about base triplet number 4 from, yes, base triplet number 4. So this is the first instance. The second instance, what will happen to a protein if the base triplet number 4, ATA, is changed to ATT? So ATA, I have to convert it because the DNA, I need them to be in messenger RNA. ATA is going to be, T goes with A, U. And then this one is going to be U. So now I say that this will change into this. Okay, this will code for this, and this will code for this. Cool. That's point number 1. But what does this code for? AAU. AAU is valine. What about UUU? UUU, is it valine? No. Where is it? Do we have UUU? No, man. It's not UU. Yeah. What am I doing? It's AAA, ne? It's AAA because T goes with A, T goes with A. So it's AAA. So what am I looking for? Is the AA is proline. So it means that if you look here, this one code for valine. valine, while this one code for proline. Proline, you see? So it means that these two, they are different. Is it clear? So it means that even the protein will change. It's like calling you Carabo, and then I call another, I call again Cabero. And Cabero is different from Carabo. You see? Yes. So it means that now this will code for this will code for this, of which this code is for valine, and then this, you write this in full, that AAA, ne, which code for proline. Therefore, a different protein will be formed because of the different amino acid. That's the scenario number 2. Scenario number 3. Yes, one scenario will come. Ne? Explain what will happen to a protein if DNA base triplet, or if DNA base has changed. Yes, there they have not talked about you have to interpret it automatically. If the DNA sequence has changed, one base has removed or has been replaced, whatever, it will lead to a different sequence of messenger, which will lead to a different sequence of transferrin A. Yes, which means a different sequence of amino acid. You understand? Hence a different protein. Different sequence of DNA will lead to a different sequence of messenger RNA, which leads to a different sequence of transferrin A with its amino acids, which leads to a different protein. That's a study scenario. The first scenario, it might not change the protein. Therefore, the protein is not going to change because the amino acids are being coded by different codes as we saw it. The second scenario, they can code for a different amino acid, meaning that valine and proline, for example, therefore a different protein. Or they can ask you what will happen if DNA base sequence changes or messenger RNA sequence changes. So you start from messenger RNA sequence. If it changes, it will lead to a different sequence of transferrin A with its amino acid, hence a different protein. Guys, don't go without knowing those things. Lastly, on DNA, it's this. Let me just use this example, but you still have questions. You still have questions we can use. Please, 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 don't go to the paper without knowing that. Someone is telling me they have given you T or C. Just in mind A and C. Yeah. So guys, make sure that you guys get the things right. Yes. If you have any challenge, let me know. Yeah. Lucy, Lucia. Yes, you're saying adenine join. Yeah, I made a small mistake, but yeah, I saw it when I was trying to. Yeah, I'm also human. I can make a mistake. Yeah, but when I do it, sometimes when you talk, you can talk about something else, and then what in mind is something different. Yeah. All right, but I'm sober today. I'm sober. I don't drink and smoke. So I'm sober. All right. All right, let's continue to the next. Guys, don't go without knowing how to label these things. What is this? It is amino acid. What is this? Is ribosome. What is this? What is this? It's transfer. You see, it is a T. Guys, check. It's a T. tRNA. If you don't know it, try to see if that T will come up. Check. You see? It comes out. It's a T. So it's a T transferrin. Guys, this is not a protein. It's a polypeptide chain. Protein is 50 amino acid and above. Is it clear? So this is DNA molecule. This is messenger RNA. This is nuclear pore. This is the nuclear membrane. Please go when you know how to label these simple things. Is it clear? Yes. This is the transcription. It occurs in the nucleus. This is translation. It occurs in the cytoplasm. Therefore, if they ask you what is this space, is cytoplasm, and what is this space, is the nucleus. Yes. Lastly, but not the least on DNA, so that we go to meiosis. Two things on here. The first thing is if DNA is 30%, if adenine is 30%, what is the ratio of guanine to cytosine? What's the ratio of guanine to thymine? Let me just say like that. Guys, you have to know how to do the base calculation. A goes with the T, C goes with the G. If this is 30%, automatically also this is 30%. So 30 plus 30 gives me 60. So remember, this is percentage, né? Percentage means 100. So 100 minus 60, it gives me 40%. So this 40% is for these two. So divide by 2, so it's going to be 20, it's going to be 20. That's not the answer. They're saying the ratio. So it's going to be G to T, whereby G, where's G? G is 20, and then T is 30. So I divide by 10, I divide by 10, so I get 2 here, and then I get 3 here. You know what we're going to do? The first objective we're going to do, we're going to say that 3 to 2. Because when you saw 3 and 2, you just say that it's A. And the last one is going to be 2 to 3, because you don't want to read everything. Make sure that you read everything, because there is most correct answer. I remember in paper 1, whereby they gave a correct answer halfway, correct? And there is the most correct answer just down. You understand? We always try to trick you to see that, yeah, you are off. But I know since you are here, you won't be off. Ne? Yeah, we are here to kill the paper, not the paper to kill us. Ne? Yeah, tomorrow we will make it. We will make it. We'll make it. I know. All right. Yeah, if do you have any question, let me know. Let me know if you have any question. Let me know so that I smash that question without. That's number one. That's number one. The last thing which I need to talk about is about profiling. This profiling. Someone might say that, profiling is easy, man. Yeah, I know that profiling is easy, man, but there is a time when they are talking about comparing the paternity test. Ne? Let me see if this question is about paternity test. Identifying the criminal, or what is this? Okay. Sample DNA sample. Okay, let me just read it. Detective investigating the scene of broken window, suspected blood was found in the criminal identified. No, it won't give me what I want. Blood. And no, it won't give me what I want. Okay, now let me design my question so that now you understand what I'm saying. If they say that two guys, they are crying, or they are saying that, okay, you have a boy, and then you have two guys, ne? And these guys, they are saying that it's each is saying that is the father of this boy. But now the mother doesn't know who, because this mother slept with these guys, ne? These guys are the same day, ne? You understand? So now we don't know who is the father of this boy. So what you need to do is, if this is the DNA profile of the boy, guys, listen until we end this, ne? Because sometimes this question, when it comes, you are off, ne? Okay, if this is the profile of the child, yes, this the profile of the child. I know DNA profile is very easy, but once it comes to this, you lose the marks. Yes, you lose the marks. Okay, this is the boy, ne? And then now you have the profile of the mother. You have the what? The profile of the what? Of the mother. You have the profile of the mother. Is it clear? Yes, you have the profile of the what? Of the mother there. And then now you have the first profile of the guy. Well, the first profile of the guy. You have the first profile of the first guy. You have the first profile of the first guy. Ne? Of the first profile of the first guy. Yes. And then you have the second profile of the second guy. You have the first, you have another profile of the second guy. You have another profile of the second guy. Yes. Yeah. So now they ask you, this is the mother, this is X, this is Y. Is it clear? Who is the father of this guy? And after that, they can ask you to explain, and they say 4 marks. Explain 4 marks. That's it. That's what I'm saying, that's where students go off when it comes to this. Listen carefully what you're supposed to say. Why did I divide with the 10 so that I get the simplest ratio? Ne? Yes, I divided by 10 so that I can get the simplest ratio. Yes. To simplify the ratio, yeah. Holy. Yes, correct. What you said is correct. I divided by 10 so that I can get the simplest ratio. Equilibrium, I'll reach there, don't worry. So, guys, look here critically. What you need to do here is to first, because I know the mother. So take all the bars which correspond with the mother. So this corresponds with the mother. Yes. This bar correspond with the mother. Yes. This bar correspond with the mother. So it means that, let me just use a different color so that we see exactly. So this bar is out, this bar is out, this bar is out. So now the remaining bars, not that one. I'm saying this bar is out with the mother, this bar out with the mother, and then this bar is out with the mother. So the remaining bars, these remaining bars, this, and this, it must correspond with one of the guy. If there is one bar which is not corresponding, then that's not a father. Is it clear? Yes. Now I come here, I look for this. What does this one correspond with? This. If there is no one who is corresponding with it, then it's out. You understand? Then is what? Is out. So this bar correspond with the, with this, ne? Yes. So it corresponds with that. This one is a little bit down. And then this bar corresponds with that. This bar corresponds with this and this. So however much this one has one which is corresponding, but this one and this one did not match with it. So the remaining bars, you see that all the remaining bars, yes, have corresponded with this guy X. Therefore, guy X is the father. Explain your answer. Why are you saying that X is the father? X is the father because the bars of the boy correspond, the remaining bars which of the boy which do not correspond with the mother correspond with this guy X. All the remaining, the word all must be there. All the remaining bars of the boy, yes, after matching them with the mother, correspond with guy X. Is it clear? Because they always ask you this. Yes. And then students tend to go off. These bars must correspond with the mother. The remaining bars must correspond with the father. If there's one bar which is not corresponding, we will ask you, where did that boy get that gene? Understand? Remember, we get these chromosomes as a sperm and ovum. Where did that boy get that gene from? Are trying to mean there's a mutation? So that's why all of them must correspond with the father, so that we conclude that that gentleman X is the father of that boy. Yeah, guys, unless you have any question concerning about describe the process of translation according to the messenger RNA, the transfer RNA with the complementary anticodon match according to the messenger RNA, the transfer RNA with the complementary anticodon. Yes, bring the required amino acid to the ribosome. The amino acid is joined together by a peptide bond to form a required protein. Guys, those are just processes I'm not going to go through. Maybe I can ask a question. What is DNA replication? When does it occur? When does it occur? When does it occur? It occurs during before the cell divides, during interphase. Where does it occur in the nucleus? Why does it occur to double the chromosome number? Yes. How does it occur? That is the process of DNA replication. So there are so many questions. I'm not going to go through them, but make sure that you know those processes. Unless you have a question about DNA replication, about DNA code of life, let's go to meiosis. Yes, when we talk about meiosis, guys, there are some things which you need to talk about. See, this is a transfer. Okay? Makes it. You see, this is amino acid. This is transfer. We said it. This is a ribosome. This is messenger RNA. Sometimes it's up, sometimes it's down. This is what? Codons. Hey guys, how many? This question. Two questions. The first one is how many codons in 12 bases. So remember, 3 bases is equal to 1 codon. So it means that 12 divided by 3. So makes them to be 4 codons. How many codon? How many bases? How many amino acids in the 4 codons? It's gonna be 4 because each codon code for 1 amino acid. Understand? How many bases code for 5 amino acid? Remember, each amino acid is coded by 3 bases. So I'm gonna say 5 times 3, which gives me 15. You understand? So you have to know how to interpret that. And then, let's go to meiosis. Let's go to meiosis. Yeah, we try to explain this. They say left, right. Make sure that you translate it left, right. Ne? Yes. And the first thing you need to know about meiosis are the phases. But I'm not going to go through the phases. Use mat, you'll be able to know which phase is it. But guys, something is itching me. And that itch makes people to lose marks in meiosis. Number 1, identify phase in diagram 2, diagram 1, diagram 3, and diagram 2. Guys, whenever they ask you about the phase, please turn your paper if these centromere, the centrioles, they are not on the sides. So turn your paper to be like, yes, you see, it must be, I don't know, I think this is east and this is west. If I'm not a geographer, I hope this is the correct. Yeah, but it must move east to west, east to west. It must leave what? East to west. Don't leave this. They must be in east to west so that you identify the perfect what. If it's not straight with a paper, turn it. Turn it, because if you turn it, you will be able to identify the actual what? The actual phase. So in this case, because there is space here, it means that is anaphase. But because it is single, therefore is going to be anaphase 2. So it's anaphase 2. What about here? The chromosome, you see there in the equator. So it's going to be metaphase 1. Yes, because the chromosomes are double, you see, therefore it's going to be metaphase 1. If they're single, then it's going to be meiosis 2, which is you identify the phase and then you put the word 2. That is the first thing which you need to know before you go for the paper. The second thing which you need to know about, before I tell you what to focus more on this, is the number of chromosomes. How many chromosomes are found in diagram 1? You will find out someone saying 46. Did they say this is a human cell? No. If they don't talk about human cell, don't talk about 46, don't talk about 23, because they didn't talk about human cell. How many do you see? 1, 2, 3, 4. It's 4. How many chromatids found in diagram 1? It's 1, 2, 3, 4, 5, 6, 7, 8. It's 8. Chromatids they're talking about. How many chromosomes will be found at the end of division of diagram 2? Yes, because this is meiosis 1, therefore it's gonna be, since there are 4, therefore it's gonna be 2. Yes, that's the number of chromosomes will be found at the end of meiosis 1. Now the question comes, how many chromosomes will be found in diagram 2 if this was a human cell? Now, they said, if this was a human cell, because this is meiosis 1, human cell, therefore there will be 46, or there will be 23 pairs. Yes, I think I'm clear there. That is point number 1 about meiosis. Point number 2 is about the karyotype. This is where also you lose marks. Describe the structure, describe the karyotype. It's fine. You can describe that from this karyotype is for a male. If you see this is for a male, this is for a female. Because here, the chromosome number 23, they have the same size and shape. While this one is big, one is small. Yes, therefore it means one is X, one is Y. But now, if they ask you to describe the karyotype, from it's made up of 46 chromosomes, which are arranged in pairs. From pair number 1 to pair number 22, we call them orosomes. The 23rd pair is called the gonosome. The gonosome has X and Y. If it is a male, has X and X. If it is a female, the X is longer than Y. So you describe what you see. But if they ask you, how many autosomes are found in P? Please, guys, don't just say that is 23. Sorry, don't just say that is 40, 40, 44, or is 22 pairs. No, count them, because sometimes maybe chromosome number 6, we put them 3, whereby you say 1, 2, 3, 4, and you don't see that, you don't recognize. Look at chromosome number 21, but it's better to count them 1, 2, 3, 4, 5, because you'll find out. You won't just ask you a question, how many chromosomes are there? No, man, they will have to hide something. There must be a reason why they ask you that. Ne? Yes, there must be a reason why they ask you that. Maybe there is a chromosome which they have they have done the hiding, and then when you have to identify where they have hide that. Guys, give it or don't panic. Take your time, read the question 3 times exactly. If you feel like you don't understand, skip it. Yes, why right? What? Start, if you see that things are not working out, start with what you think is right. I'm telling you, don't panic. Don't panic because you have not seen the diagram, Yeah, the diagram is known. This time they have brought it in terms of what? So why are you skipping? Skipping what? I'm not skipping anything. Yeah, I'm going to come back. I'm trying to talk about the things. After that, I come back to those questions. Ne? Yes. So guys, make sure that you concentrate on those karyotype. Make sure that you know how to identify them. Give a reason. Give one observable reason why state the gender of P. P, where is P? This one. We say that this P is what? Is a what? Is a male. Why? Because the chromosome number, the pair number 23, it has 2 different chromosomes. One is X, one is Y, or one is big, one is small, meaning that one is X, one is Y. Yes, that's number 2. That's where people fail. Number 3, non-disjunction. Non-disjunction. You see this? This is a non-disjunction question. Yes. Whereby when they ask you to which phase is it, is anaphase. It could be anaphase 1 or anaphase 2. Yes, but looking at the diagram, you can identify that it's anaphase 1 or anaphase 2. Yes. Describe Down syndrome. How is it? How is non-disjunction lead to Down syndrome? Guys, you just talk about what is extra. That Down syndrome is as a result of non-disjunction, leading to one cell having an extra chromosome. Yes, on chromosome number 21. Yes, meaning that it's gonna have 24 chromosomes instead of 23. Yes. When this cell fertilizes a normal ovum, the resultant individual is going to have 47 chromosomes instead of 46, leading to Down syndrome. Is it clear? Down syndrome is caused by non-disjunction. Is it clear? Either in anaphase, it could be anaphase 1 or anaphase 2, leading to a cell having an extra chromosome on chromosome number 21. In brackets, you can say 24 chromosomes instead of 23. Then, when this gamete, yes, fuses a normal gamete, meaning that it is having 23, it will result in 46 chromosome, a zygote with the 46 chromosome, 47 chromosome instead of 46, leading to Down syndrome. It's simple. I don't know why people make it difficult and complex and what. No, man, it's simple. Is it clear? Yes. So that is another thing where you lose marks. Number 3, give 2. Let me check here. I hope that question is here. Metaphase 1 and anaphase 1. Those ones you can do. Okay. Describe 2 ways in which this is Down syndrome. Yes. So we have answered that one. Yes. Name the organ in female that where meiosis occur. Okay, meiosis occurs in the testes for the case of male, and then female occurs in the ovary to produce ovum. Yes, but there is something which I want where students fail. Give 2 causes of variation in meiosis. Guys, they are saying in meiosis. in meiosis. They're not talking about the causes of variation. No, they're saying about in meiosis. So what are you supposed to talk about here? You talk about the the random arrangement of chromosomes during metaphase. Yes, random arrangement of chromosomes during metaphase, and also talking about crossing over during prophase 1. Remember, when you talk about prophase, crossing over, you are specific to prophase 1. Prophase 1, not anything else, is prophase 1. You don't talk about something else. It's supposed to be prophase 1. That is crossing over during prophase 1, which leads to exchange of the genetic material. So please don't go to the paper without knowing the process of crossing over. Don't go to the paper without knowing anaphase. Don't go to the paper without knowing metaphase. Yes, metaphase 1, metaphase 2, anaphase 1, anaphase 2. Those we might likely to bring it. Division of a cytoplasm, what does it mean? Is called cytokinesis. This division of a cytoplasm is called cytokinesis. Lastly, on meiosis, what you can't go without knowing is know how to label. What is this? In most cases, people, they call this nuclear membrane. No, nuclear membrane cannot be seen. You see, the nuclear membrane can only be seen in telophase. Yes, yes, in telophase. And in prophase, prophase. So this is cell membrane. This is chromatid. This is centriole. The dot is centromere. This is spindle fiber. Ne? This is, what is this? This is a cytoplasm. So now I think you know. The last question whereby students get lost is draw the diagram of a cell at the end of meiosis, or draw the diagram of a cell after crossing over. Let me just show you. Let me just show you using this. Using this, I think I have the diagram here. Give the collective name, give the gender, name of observable reason, the main result of segregation, main result of segregation, describe the karyotype, and syndrome will result differ. Okay, sure. That's not what I want. Yes, here. Look at that diagram. Look at that diagram, guys. If they ask you to draw the cell or this after this process, guys, look at where crossing over took place. In this case, the crossing over took place up. While here is took place here and here. So if they ask you to draw this cell, maybe they are saying chromosome X and chromosome Y, whereby you say structure of chromosome Y after maybe crossing over, then you have to draw it nicely. You have to draw it nicely. Yes, you have to draw it nicely. But where did the crossing over take place? Crossing over took place on this chromatid, but halfway up. So it means that when I'm shading all this, remember this is the black thing, so I have to shade all this. I have to shade all this. Yes, I have to shade all this to be black. And then the crossing over did not start from here, it started from here. So this variation of shading will give you a mark. Writing a caption will give you a mark. And then don't forget to label. Even if they have not asked you to label, label. Even if you don't ask you to label, at least have 2. Because in most cases, when they ask you to label, at least 2 points. Yes, they say at least 2 points. You have labeled it at least 2 points. At least then you get those marks. Ne? Yeah, please don't forget to label. If they don't say, if they say draw a label diagram, or they'll say draw and then just leave it like that. Guys, make sure that you draw and label, because it's not losing you. It's not taking, they won't cross it because it's labeled. But you get a mark if you label, just in case, because you are not a memorandum to know which one is not going to be credited or is not going to be, is going to be credited. So you just write. Lastly, before I forget, if they ask you to describe, write everything you know in the head. At least for us, we look for what is correct for you. If they say name, that one you only name. You don't give alternatives. But if they say describe, just write everything you know. Write everything you know. Or sometimes I call it write all the rubbish. I'm not saying that write the rubbish, but write everything, whatever you have in the head. Don't leave it, because when you leave it, you come out of the paper and say, I thought about it. I thought about it. I thought about it. No, don't do the regrets, because this is your last time to change this subject, the mark. So just write everything. If the correct answer is there, we look for it and we'll give you a tip. Ne? Yes. Yeah. Do you have any question, guys? Comparative anatomy. Someone saying comparative anatomy. Okay. Thank you. Guys, if you have any question, you let me know, né? Because I'm here to make sure that you get a distinction. You get a what? A distinction. Distinction. Yeah. Make sure that the distinction clicks because we want it and we will get it. We will get it. Yes. God willing, you know me, when in my region we say, Inshallah, Inshallah, we will get a distinction. God willing, we will get a distinction. That's what it means. All right. Check this. Check this. It's simple. You guys, I'm winding up now. Ne? Make sure that you know how to label those diagrams. Make sure that you go when you know the process of crossing over. Make sure that you know the karyotype. Make sure that you know the prophase, anaphase, telophase. Make sure that you know when you go there, when they give you a diagram, it must be east to west. Those spindle fibers, they must be east to west, meaning that you have to turn the paper. Turn the paper. No one is going to ask you, why are you turning the paper? It's your paper. No one is going to ask you whether you turn it, whether you write upside down. Doesn't matter. Turn it so that you identify the things correctly. You understand? Yeah, these phases, they are confusing people to identify. Please don't make a mistake. It's the last time for you to identify what is it. Let's try to label this and then you see. How do you call C? Is the centromere. How do you call this? The point where crossing over takes place is chiasma. How do you call this? We call it chromatid. How do you call this? Because they are chromosomes and crossing over, we call it bivalent. Yes, we call it a bivalent or bivalent. Yes. Or you can call them, sometimes you can call them, they can correct you when you say homologous chromosomes. Yeah, but scientifically we call it a bivalent. Bivalent, né? Or bivalent. Né? Someone might ask me the spelling. Yes. So identify, okay, we have done that. Name two ways in which structure A and B is similar. Structure A and B. How are they similar? How are these two structures similar? They have the same size, né? Same size and shape. Good. Number two, they have the same location of the chromosomes. Not same chromosome. They are not the same chromosome. If they're same chromosome, you'd have looked exactly the same as your father. Yes. So they have the same position, locus, né? Same position of the genes. They have the same position of the genes. Describe the process at D. So they are looking at describe the process of crossing over 5 marks. The homologous chromosome come together. They cross over at a point called chiasma. They exchange the genetic material between two homologous chromatids, and then they separate, and then they form genetically different chromosomes. So basically, that's what you need to explain when it comes to that. I'm not going to use too much of the distinction material, but I know that you guys, you will make it. Lastly, before I go, where does meiosis occur in male? It occurs in the ovary to form ovum, testis to form spermatozoa, then in plants, ovary to form ovules, anthers to form what? Grain. That's it. Hey guys, I feel good when you guys are feeling good. Ne? Don't panic, ne? Because when you panic, you make me panic. Ne? Yes. So make sure that you don't panic. Ne? Yes. Make sure I repeat. Don't panic. You're gonna make it. Let's go to genetics. Genetics. What I'm supposed to talk about genetics. Genetics. So we have used 1 full hour. Remember, we said that we're gonna discuss this paper for 2 hours. Ne? So we have used 1 full hour. So let's go to the second hour, that is genetics, and 30 minutes, and then 30 minutes for evolution. And then we know that we have smashed. Guys, make sure that you get all the marks in DNA, all the marks in meiosis. Yes, guys, in genetics, we'll talk about genetics. Terminologies, you know, I'm not going to talk about the terminologies. But go when you know that a circle means a female, and then a box means a male. Is it clear? That's number 1. Number 2, when they give you a question, identify whether it's a sex linked or a non-sex linked. I repeat, identify whether it's a sex linked or a non-sex linked. How do I differentiate between the two? Sex linked, they will mention it that is sex linked disorder. Always read what is on top here. Is a sex linked disorder. Is it clear? Make sure that you read it. If they say sex linked disorder, definitely you have to use X and Y. Male, there will be X and Y. Female, there will be X and X. If it is not a sex linked disorder, please don't use X and Y. However much they have talked about same and carabo, and you know that this is a male, this is a female. Please don't use X and Y if they have not mentioned that this is a sex-linked disorder. I think I'm clear there. Number 2. Let's do one example on sex-linked recessive allele. One example, sex-linked dominant allele. One example, non-sex-linked. The first one, identify whether it's a sex-linked or non-sex-linked. Okay, now we have identified here that is sex-linked. The second thing you need to know, guys, I repeat, and I'm putting down the pen, the mouse. I wish you could see me the way I'm begging. Please, let me come closer to the mic. Please identify whether is caused by a dominant allele or a recessive allele. I repeat, identify whether it's caused by a dominant allele or recessive allele. Because if it is caused by a recessive allele, the one who is homozygous recessive is the one who is going to be affected. If it is caused by a dominant allele, the one who is homozygous recessive is going to be normal. Let me show you using here. This diagram is saying. So it means that it's caused by a recessive allele. Ne? They said it here. Ne? So it means that the one who is shaded, they're saying that is affected with the disease. Therefore, this person is a male because the sex linked is going to be that way. While this is a female, is going to be small d, small d. Is it clear? This one is a male, is going to be capital D, Y. Okay. Question. What about if it's caused by a dominant allele? Perfect. This is caused by a dominant allele. Yes. Okay, let me first finish here so that I don't confuse you. Now, let me go to the questions and see how many generations are shown here. It's 1, it's 2, it's 3. So there are 3 generations. How many kids listen carefully, how many kids are sons of 1 and 2? How many kids are sons of 1 and 2? 0. How many kids are 1 and 2? 2. Remember that this is not a kid of this. This is just came and married this daughter. And this one came and married that daughter. Therefore, there are how many? There are 2 kids which are coming from these 2. Is it clear? You see that this thing is not linking down. Is it clear? Yes. I'm done with that. Let's go to the third question. How many family members are in this phylogenetic tree? Family members. It's 1, it's 2, 3, 4, 5, 6, 7, 8, 9, 10. They are saying family members. Is it clear? They're saying family members. Don't say that this is not a family member, unless you're telling me that your father is not among the family. It's a family member. But if they say how many kids for this, then I think now it's clear you know what you're supposed to do. Yes. That's the third thing. If they say that this cross with this, produce these kids, do a genetic cross, because we are in the second generation, so it's going to be P2 F2. Is it clear? If they say this, it's going to be P1, F1. I think I'm clear there. Yes. So I will talk about when I go to the what? The crosses. Then we go to the next, what you're supposed to know. Yes. Can you please do a live letter? Today I join. Can you do the live again later today? I join. No, what you can do, if the live is start from where you have started from, and then after that, join. You will play the video. Ne? Yes, before. Lindo, Putle. Why are you removing? I didn't see what you are saying, sir. The sound at the back. Eh, someone saying there's a sound at the back. Ah, serious, there's a sound at the back. Okay, let's go to the next question. Okay, how do I find the unknown one? I'm gonna talk about it, my daughter. Um, yeah, guys, I hope the sound at the back is removed. Let me know. Let me know. Let me know if the echo sound is removed. I don't want those echo sounds. Yeah, it must be nice. All right, let's go to the next question. How do I identify? Okay, now someone is asking. I'm gonna go there so that we finish this. How do I identify one? To identify one, remember you have to first identify that this is caused by a recessive allele. Yes, if it's caused by a recessive allele, so what you need to do is okay, what you need to do is, okay, this one is XX, but this person is normal. Therefore, it's going to be capital D. I don't know whether it's going to be a capital D, small d, or capital D, capital D. But they produced a kid who is sick. Therefore, this kid is sick with a, don't just say sick, sick with what? With the disease. So automatically, one came from the father, one came from the mother. Therefore, this mother cannot be like this because it gave rise to this. You see? Whenever they ask you to identify, make sure that you look at the kids. Yes, which kind of kids they have produced. Therefore, we have identified that because this kid is small d, therefore one came from the mother, so therefore the mother is like this. Now what about the father? Remember, this is sex-linked disorder. Yes, if one came from the mother, then definitely another one came from the father. Therefore the second x, the first x is like this, or the x is like that. Therefore the father is affected with that disorder. Don't just say affected. Affected with the what? HIV, sickle cell, flu, corona is affected with what? With the condition they have talked about. Don't leave the answer hanging. Please, 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 please. Don't leave the answer hanging. Then the second one. Okay, someone saying, ish. Hey guys, just give me 2 minutes. Let me check why. Why is it? Ahem. I hope the sound is gone. I don't know whether. Okay. I hope the sound is gone. Yeah, you let me know. Please slow down. I hope. I hope it's okay now. All right, let's continue, guys. Let me know if the sound is fine. I don't know because I've tried to see that everything is right. I've tried to check. Let me check. Let me check here. I've tried to see that. Yes. Let's continue and see. Ahem. Hey guys, I think that sound, I don't know, because we changed the system and the system I'm using is a different one from the main one. Yes. Yes. So I had to change the stations. All right. Let me just continue. I hope at least you get something. Yes. Okay. I think now you have got it correctly. How do we identify this? Yes. Now let's go to the question, the next question. Then we are finding out. Identify the phenotype of individual two. These ones I'm going to do the questions and then we see. The phenotype of individual two. So we have said that this person is normal. Yes, is normal. A normal male or female is a normal female or female without C. Yeah, that disorder. Then the phenotype of one, we have identified that this person, because they have produced a kid who is sick and the sick is like this, therefore one recessive allele came from here, another recessive allele came from here. And remember, this is a male. This is a male. Therefore, this individual is affected with SID. And then they're saying that identify the genotype of number 3. Genotype of number 3, yes. Remember, this person is sick. Therefore, is going to give remember, is a female. It's going to give one chromosome from there. But because it's normal, therefore the mother gave her this. Therefore, it's going to be that way. Because they say the genotype. They say explain how individual 5 inherited disorder. Individual 5. Individual 5 is small d. Therefore, we're going to say that individual 5 got one recessive allele from the mother and then one recessive allele from the father. No. Individual 5 is a male, is a male. Can't be from the father. Father is normal. The individual is male. So we're gonna say that the individual 5 is a male who is sick and then therefore obtained a recessive allele from the mother. Where is these 2 marks? You have to tell us that individual 5 is affected with this disorder and is received one recessive allele from the mother who is number 4. Therefore, I think that's how you can try to explain. But now let's go to A. Guys, the problem comes in when it is caused by a dominant allele. Okay, let's look at it. When it's caused by a dominant allele. Puti, whatever, is syndrome sex-linked. You see, they have talked about it, but it's caused by a dominant allele, isn't it? The diagram below shows this syndrome in the family. Okay, now we are stating how many generations are there? There are 3. 1, 2, 3. Sharp. How many females are in this phylogenetic tree or in this family? Females. 1, 2, 3, 4, 5. So 1, 2, 3, 4, 5. Is it clear? How many females were produced by Patty and Gabby? It's gonna be females. 1, 2. This one is not family member, not a kid of this. It's a family member, but it's not a kid of this. So it's only these ones who are the kids. I think I'm clear there. Then now we go to the next question, whereby they're asking you. They're asking you. They're saying that. They're saying that. They're saying that. Okay. Name the. All right. Let's find out. Remember now they're saying that this is affected, né? And then this is also affected. This is unaffected. But don't forget that this syndrome is caused by a dominant allele. Therefore, these people who are affected, they're going to be dominant. Capital D. Can it be capital D, or capital D? Sorry. X, capital D, X. Is it capital D? No. They used G. Sorry, guys. They used G. Please use the letter they have given you. Don't just use what you think is correct. Therefore, it's going to be X, capital G, X, capital G, X, small g, no, capital G, and X, small g. Yes. But now I need to find out. We have, what about this one? This one is normal. So if it's normal, therefore it's going to be small g. It's going to be small g. Therefore, this person can't be this because this person, the kid, has a small g. Therefore, came from here. Therefore, I have to say that this individual is like this. Capital, small g. No. Capital G, small g. So that one is done. What about the female? The male. Remember, this is normal. If it's normal, therefore automatically is having small g. That's why this and this, they combine to produce a kid who is a female with the 2 recessive alleles. We are done with this. What about this? Is a female who is sick. Who is sick. Therefore, this female can't be homozygous dominant. It's going to be capital G and then small g. Why? Because one X came from here, then another G came from there. You understand? Yes. Now, how many, name the type of diagram is phylogenetic tree. They're saying how many female in this family? We talked about them. 1, 2, 3, 4, 5, 6. Male in this generation have, how many male in this generation 1 have what is syndrome? Generation 1. Where is the generation? The first generation. The first generation. Remember, this is a generation 3. 1, 2, 3. Because they are looking for male in generation 1. So this 1, 2, 3. They're talking about generation 1. They're saying with the disease. 0. Answer is 0. Because it's caused by a dominant allele. And then they're saying that give the male in generation 1 have what is disease. Give the genotype of Gabi. We talked about it because they produce a kid who is not sick. Guys, please take note which one is dominant, caused by a dominant allele, which one is caused by a recessive allele. They will have to specify that is caused by a dominant allele. Is it clear? Males in okay, we're done with that. Aju and Pirusa have another 4th child. Give a phenotype of their sons. And Pirusa. Yes, so of their sons. Remember, this is caused by a dominant allele. The moment is caused by dominant allele is easier to find. For the case of what? The sun. And then, remember this one, sun, sun. Ne? It's a Y, it's a Y. Therefore, because this one is normal, remember is recessive, is normal, therefore this one is going to be, both of them are going to be normal. Don't forget it's caused by a dominant allele. If it was a recessive allele, then they would have been affected. But the one who has the recessive allele is the one who is normal. And the one who has a dominant allele is the one who is affected with that disease. Is it clear? Yes, that is the only trick you need to know about that. The one is normal. Yes, the one who is normal is having a recessive allele for the dominant allele, if it's caused by a dominant allele. The one who is sick is the one who is having a dominant allele. You understand? Yes. Explain your answer above. Why? Because they are normal. Because Anju, yeah, who is a female, is normal with the 2 recessive alleles. Therefore, it gives 1 recessive allele to the males. You understand? Yes. Therefore, they are normal. So basically, that's what you need to talk about. When you go to a cross which involves an autosome, a non-sex linked, ne? Yes. Please, even if they talk about same and Carao, please don't use X and Y. Ne? Yeah. If they say that this also is caused by allele R, so you just say R. Ne? It is homozygous. If it's heterozygous, is R, small r. You don't put them on X and Y. That is the major difference between the two. Yes. Okay, lastly, about blood group. Then after that, we talk about the cloning. How many blood groups, how many genes control blood group? How many genes control blood group? How many genes control blood group? Someone is asking me, sir, it started confusing you. Daily interesting videos. How am I confusing you? Let me know. Let me know. Huh? How am I confusing you? Oh, where are you confused? Yeah, in this video description. You're saying I'm confusing. Let me know where am I confusing you so that I can sort that out. Ne? Yeah, just tell me that maybe this question or this question. Yeah, so that I can sort it out. How many genes? Yeah, correct. Correct, guys. Correct. Thank you very much. Yes. How many genes control blood group? It's 1. It means that you guys watch my videos. Thank you very much. And I'm seeing live as Mbali, as a member. Yeah. Thank you very much, Mbali. Yes, for being a member. And I saw another member here coming. Oh, Lindy Wayne Love, thank you very much also for joining us. So you are on the team, ne? Yes. All right. Never be saying that X and X are. Oh, okay. Okay, let me explain to you what I meant there. Someone is saying that X and XR. I'm saying don't use X and okay. Let me just look for a question here. Yes, here is a question. Ne? Mbali. I think it's Mbali, ne? Yeah. Here they're saying that this syndrome in dog is caused by a dominant allele, capital H. You see? The pedigree diagram below shows the offspring produced by 2 dogs. You understand? They're not saying that this is a sex link. You understand? Yes. How many offsprings are affected by this disease? How many offsprings? You see now? Is caused by now this one is also caused by a dominant allele. Is caused, meaning that the one who is sick, né? Check here. Even here they're saying it that dogs with the disease and dogs without the disease. So these ones which have the disease, it means that they are going to be capital H, capital H, or can be capital H, small h. Even this one is going to be capital H. You see, is a male, but I'm not using X and Y. Capital H, or capital H, small h. Whenever there is a capital H, because it's caused by a dominant allele, this individual is going to be sick. You understand? Yes. So, however much is a male, I'm not using X and Y. You understand? I don't use that. Why? Because they didn't say it is caused by a sex link. Is it clear? Yes. So in this case, but I can identify, because they say that how many individuals are affected. Okay, that one is simple. But now this one is what is not affected, meaning that this individual is going to be small h, because it's caused by a dominant allele. Whenever you see a capital H, it means that that individual is what? Is sick. If they produce a kid here who is having these two, then automatically one came from the father and one came from the mother. Therefore, they can't be this, they can't be this. So this is like this, so that one H comes here, and then this one is like this, so that another H comes there. You understand? So when they give you a question and they don't talk about sex linked, please do not use, I repeat, do not use X and Y. Use the letters the way they have given them to you. Yes? You only use X and Y if the statement says that is caused by a recessive allele, is sex linked. You see, is sex linked. Even here they said it here is sex linked. Even they gave you what to use, that is carried on X chromosome. Is it clear? Yes. So this situation is caused by a dominant allele, but it can be caused by a recessive allele. So I think now I'm clear when it comes to that difference between phylogenetic tree and pedigree. Phylogenetic tree, guys, phylogenetic tree is explaining the evolutionary relationship among the members. You understand? Evolutionary relationship, like how did you evolve? But a pedigree is describing a character in a family. Is it clear? Is describing a character in a family. Yes. Okay, guys, let's go back to blood group. I think I've got it. You have got it. I'm no longer confusing you. How many genes control blood group, guys? Is one gene. Is one gene. How many genes control blood group? Is one gene. How many alleles control blood group? One gene, and that gene is blood group. Do you have any other gene? Tell me if there's any other gene. It's one gene, blood group. Yes. But how many alleles? There are 3 alleles. Yes, that is allele for A, allele for B, and allele for O. Guys, please don't make a mistake and you say allele for A like this. This is not A. This is a Roman numeral, which means 1. Allele for B is supposed to be like this. Allele for A. Don't put at here. Don't put them, make italics. Don't say allele for O like that. What is this? We will cross it. I is supposed to be like that. Is it clear? Yes. So allele for A, allele for B, and allele for O. So there are 3 alleles. Yes. I don't want to go to the distinction material to show everything. No. We go to the next part, of which the next part they're asking you, how many genotypes? Genotypes. Oh, before I go, guys, back, there's something I forgot. Before I go back, I don't know what took me off. Why are males more affected by sex-linked disorders than females? Why? Or they can say, why are males more affected by hemophilia than females? Or they can ask, why are males affected by color blindness than females? So the answer you give here, if they say 4 marks, you say that males have 1 X. Yes. If a recessive allele, for example, is capital small h, appears on X, yes, the individual is affected with the disease, whatever they have talked about. While females, they have 2X. Yes. If a recessive allele appears, for example, on one X, it can be masked by a dominant allele on another X. Therefore, the individual is normal. Males have one X, yes. If a recessive allele appears on the X, the individual is affected with that disorder they are talking about. While females they have 2X. If a recessive allele appears on one X, it can be masked by a dominant allele on another X, and the individual is normal. Sometimes they can ask you a question that why males have higher chances of becoming hemophilic than females. So guys, it's the same thing. Please, you need to know about that. Ne? Yes, you need to know about that. We go to the next question. Because they have one X. Yes, because they have one X. This question they can come for 3 marks. Ne? Yes. Is it possible for a person of blood group A marrying a person of blood group B and they produce a child of blood group O? Is it possible? Yes, it is possible. How do I know it's possible? You look at the genetics. Ne? Okay, here the question is A. Before even I go there, let me just answer this question, at least one question, so that, yeah. A man who is blood group AB and then who is heterozygous, marry blood group B, and then have a children. How many alleles control blood group? They're now talking about the alleles. Yes? Gene 1 is 1. Allele, there are 3. Genotype, there are 6. Phenotype, there are 4. Why am I using 4? You have A, B, O, and AB. A, B, A, B. Do you understand what I'm saying? A, comma, B, comma, A, B, comma, and O. Those are the 4 phenotypes. But genotypes, there are 6 because A is homozygous or heterozygous. Let me show you. The distinction material, because some people, just talking, they might not take it, and they must take it long to understand my ideas. See, you see, here is what I'm saying from the distinction material. Blood group are as a result of multiple alleles. It's a multiple allele, yes, meaning that it has more alleles, more than 2. There are 3 or more alternative form of yes, this. There are 3. All right. So phenotypes, there are 4. 1, 2, 3, 4. You understand? And then the genotypes, there are 1, 2, 3, 4, 5, 6. That's what I say. Because this is homozygous and heterozygous, for dominant, and then homozygous recessive. Yes. Then they're saying that 3 different alleles, which are this, and then 4 blood groups or types, which we call the phenotypes. No, the genotype, the phenotypes. Yes. So it's A, B, O, A, B, and then O. That's what I'm saying. A, comma, B, comma, A, B, comma, and O. So that you understand. Yes. So now we go back to our question. Describe the type of dominance that occur in the inheritance of blood group of woman B. Describe the type of dominance that occurs in the inheritance of blood group B. Now they're saying inheritance of blood group B. Check. They're saying that a man with blood group AB and a woman who is heterozygous for blood group, yes, heterozygous for blood group B, meaning that this lady it looks like this. See? Yeah, meaning that obtained one dominant allele from the father or mother and another recessive allele from the father or the mother. So it can come either from the father or mother for A, and this either father or mother, because they are not specific in that regard. Yes. Use the genetic cross to show the possible genotypes of the phenotype of their children. So you guys here, this principle works to all of them. Start with P1, F1. Please, F1. Don't just say P1 and then you leave, you finish after you say F1. You will forget this F1 and then you lose a mark. So you say P1, F1, at least we shall come here and give you a T. Is it clear? Then you say P1 phenotype, because P means parent phenotype. Yes, you tell us. The phenotype is always obtained from the question. Check here. You use, because these 3 marks, it is 6 marks. 3 marks are mahala. You understand? Use the genetic cross to show the possible genotype of phenotype of what? But what did they say? They said a man who is blood group. So you say a man of blood group AB times a woman of blood group B. You don't need to tell us heterozygous, because when you look at someone, you can't say that he's heterozygous. We just say heterozygous, homozygous, so that you know what you're gonna cross. So you're saying P1, F1, we give you a tick. Now you tell us phenotype, you tell us a man of blood group AB times a lady of blood group B. That's all. Then we give you another tick. Why? Because you have identified it, but you always get it from the question. So that's the second mark for Mahala. Then you talk about genotype. If you don't know the genotype, if the space, talk about meiosis. If you don't know the meiosis, if the space, talk about gametes. If you don't know the gametes, leave the space. Then you talk about fertilization. Don't know this fertilization, leave the space. But you shall come here and give you a mark there. So you have got 1 mark, 2 marks, 3 marks. Meiosis and fertilization, they must go together. If you know now, that's where you're going to obtain the other 3 marks. To know the genotype, because most of the cases, you people, you get lost on the genotype. When you get the genotype wrong, the remaining thing here becomes what? Becomes wrong. Someone is asking me, please, I answered that question. You have to rewind the video. Yes, I answered that question. Eh? Yes, for case of the DNA changing the triplet. Yes, I answered that question very well. Please, you have to watch it maybe when I'm done with this session. All right, guys, let's go to the next question. That is dihybrid cross. Then I show you what you do or what you focus on dihybrid cross. Dihybrid cross, we are not going to ask you to cross. No. What we might ask you about is identify the genotypes of the parent, the phenotypes, and the gametes. Ne? Yes. So, guys, you need to know first identify which one is which. Ne? But now, if they ask you what is the genotype of the parent here, for example, ne? Don't repeat the letters before you finish. For example, the genotype of this can be capital P, even capital L. Then another one is capital small p, small L. So the one which are the same, you combine them. Capital P, capital L, small l. So this becomes the genotype of the parent. I'm just trying to show you how do you identify. And if they ask you to identify the gametes from here, then it still is fine. You will say that P goes with L. P, capital L. So this one is done. Now P again with the small l, capital P, small l. So it's done. This with this now, small p, capital L. Then this with this, small p, small l. Sometimes people, what they do is not a cross. It's not a cross, but what they do this. They do this, capital P, small p, capital P, small p, capital L, small l. So they just cross with this, capital P, capital L. They cross this now again with this, capital P, small l. And now they cross this with this, small p, capital L. And then now this with that becomes small p, small l. Still, they form the same gametes. You understand? Yes. So at least you have done. Now they can ask you, they have given you the gametes. They want you to write the genotype. Yeah, that's where the story comes from. So you start with you start with one letter, capital P. Do I have any other capital letter? Yes. So you first leave it. Ne? Do I have a small letter of the same type? Small p? Yes. If I have a small p, I write the small p there. So they are supposed to be 2 letters. But if I don't have a small p, then definitely I will have to write a capital letter. Is it clear? I will have to write a capital letter here. But because I have a small p, therefore I will have to write the small p. small p. Then I go to L, capital L. Do I have another capital L? Yes. Do I have a small L? Yes, I have a small L. Therefore, it's going to go with small L. Therefore, this becomes the genotype of the parent. Check. It's the same thing. Capital P, small p, capital L, small L. Then when you come here, it's capital P, small p, capital large L, or capital L, small L. So that's how you can identify that. Difference between transcription and DNA replication, I talked about it, guys. I talked about it. Just wind up. Just wind up. I know that you will find everything you have asked me. Eh? You are trying to ask me. Yes. Lindo, yes, I talked about it. Difference between transcription and DNA replication. Yes. And even the similarities also talked about. Then that both the DNA double helix, unwind, they talk about both there is unzipping. Yes, when you talk about differences, you can talk about DNA is formed, RNA is formed, two templates are formed, one template is formed. So you guys, we talked about it, né? Yes. All right, let's continue to the next one. How many genes control? Once you see the dihybrid, then there are two what? How many genes? There are two genes. Ne? Dihybrid is controlled by two genes. Ne? Yes. How many alleles? Yeah, in most cases, there are 4 alleles. Yes, you find out the capital letter and the small letter. So depending on whether you see they are here. It's 1, 2, 3, 4. But there are two genes. Genes for the number of seeds per pod and then the shape of the what? The leaf. So guys, the hybrid is gonna be is simple, né? Simple. Just make sure that you do it. Né? Yes. So it's the same thing here. No one is gonna ask you to cross. Before I leave this point, I wanna ask you a question. Is it conclusive to use blood group to identify the paternity, the paternity of the individual? Is it conclusive? I want yes or no. Is it conclusive? Then once I see the answer, then I will give the reason. If the answer is correct, I will give a reason why. Three factors of, okay, the experiment, that's one you must know, guys. Lastly, let's go to, before I go to, please read about the stem cells, read about the cloning, yes, cloning, the process of cloning. I designated a full video on cloning. Stem cells, what are stem cells? The cells which have ability to modify and form other body structures. Yeah, basically, you just need to read this and the examples of these stem cells. Yes, let me just show you here from Excel and Life Science Distinction Material. Yeah, see sources of bone marrow and umbilical cord, adipose tissues, amniotic fluid, uses, yeah, to generate new or replace the worn out cell or damaged cells, or to generate new cells, be used for research purposes. Yeah, so cloning, please know how to use this cloning. But most especially, let me just talk about, yeah, it can come like this or it can come as a doll. Doll what? Do you know that this is the name of a person? It's a singer. Dolly is a singer from, I think it's from America. Do you know that? That is a singer from America. You just listen to people's songs, but you don't know that they're asking you questions and you don't know. I hope he's a singer, eh? I'm not a fan of. Okay, is it conclude? Okay, thank you very much, guys. Everyone is saying, sir, can you please touch on stem cells? Okay, sure. Someone is yes, I'm saying that. No. Yes, it's no. It's not conclusive because many people share the same type of bloody group. Therefore, it's not conclusive. It's not conclusive. Yes, it's not conclusive. Someone say, yes, I'm going to talk about them. Let me just talk about stem cells, then we shall go to, not stem cells, I'm done with stem cells. We talk about the cloning. Guys, this is what I want to clone, is this. I want to clone, let me just go like this so that it gives me the full version. I want to clone this. Therefore, I have these two. I get ship A. If I want to clone this, I have to keep its nucleus. Is it clear? Why? Because the nucleus contains the genetic material. So I keep the nucleus, I throw away the shell. Then this one, I remove the nucleus because I don't want its genetic material, but I use the gamete, the ovum. But here I just get any cell from anywhere else apart from a gamete. So I throw away the nucleus because I don't want it, but now I keep this. But why do I need to have, why do I have to be with a casing of this cell? Why should I be with the casing? Yes, it's like you drinking water in a bottle of alcohol. What are we going to say? We shall say that you're drinking alcohol, Baba. Yes, that's what you're going to say, that you're drinking alcohol. Yes. Why? Because you are putting water in a bottle of alcohol. So when I put this nucleus into a cell, yes, this cell, it will look like a gamete. Remember, this is a diploid cell from anything else. So if I put this nucleus to this, then this one is going to look like a zygote because this is a casing of an ovum. And I've put a nucleus which is 2N. You understand? Therefore, it's going to look like 2N. So if they ask you how many chromosomes are here, these are, if they say the sheep has 24, therefore it's going to be 24, or it's 2N. Yes, you understand? Yes. So now, we are saying that which method do we use? We also ask you about the method, is mild electric shock. Then you have here the foster mother here. And then we bring this, we put it in here. And then after putting it here, then this one starts to develop, develop that embryo, and then it forms the baby. This baby is exactly the same as this. You can ask you a question, why do we remove the nucleus? Why do we keep the nucleus? Because we wanted the genetic information from sheep 1. Why did we throw the nucleus away? Because we wanted the casing of the ovum. We are not interested in the genetics here. Yes. So which method we use? Mild electric shock. And then which sheep will this, which of these two sheep will this one resemble? Sheep 1. Give a reason for your answer, because we used its the nucleus, we used its nucleus or the genetic material to produce the sheep. Yes. So when they ask you such questions, they're not that difficult. You'll be able to answer and get it right. Paternity test, blood group. So it's the same thing we are saying. This is the question I was asking. Is it conclusive? It's not conclusive because many have the same type of blood group. So it's not conclusive to confirm or to determine that this is, yes, the parent of the child. Guys, now we are going to evolution. Yes. Guys, before I start to talk about evolution, the way now we are setting is a little bit different. We used to have a lot of marks in human evolution, and we are specific that human evolution takes this amount of marks. But now it's not the case. Why? We can have more marks in evolution by natural selection, and that's what they usually do now. I don't know why. And the more they bring more questions in evolution by natural selection, the paper becomes harder. Becomes harder because they talk about, we only have few sections to set in evolution by natural selection, but they try to tilt the thing. So today I want to tell you that, listen to what I'm going to say. Be ready. Show them that you are not here to joke. You're going to get a distinction. You're going to get what? A distinction, and you're going to be set free to this world. All right, let's start. Why don't we use the whole genetic material? Why? So why don't we use the whole genetic material of sheep 1. Eh? So why don't we use the whole genetic material of sheep 1? Oh, why don't we, maybe you are asking me, why don't we use the whole cell of sheep 1? Ne? Yeah, it's because this casing is not for ovum. This casing, the casing is not for ovum. You understand? And so therefore it cannot undergo meiosis. It cannot not undergo mitosis too. It won't look like ne? It won't look like a zygote, yes? Yeah, because it is coming from this other body cell. It's not coming from the gamete. That's why we have to bring a nucleus, its genetic material, and then we put it in the casing of the ovum so that this thing looks like a zygote. Understand? It must look like a zygote so that when we put it in the womb doesn't know what is inside the what? This zygote-like structure, yes? What it knows is this, né? So that now it can start undergoing mitosis and then forms those finger-like projection, and then done. Implantation takes place. So that part of implantation is paper 1, eh? So we are done with that. So that's why we don't use the whole thing. If you bring this, then you put it in, it won't be recognized as a zygote. It won't be recognized. It will be recognized as any body cell. Eh? Yes. Scientist. Let's just do 2, 3, 4 questions here, and then we go to human evolution, and then I wind up. Scientist, let me see how many minutes we have. We have 30 minutes. We have 30 minutes. We are saying that scientists hypothesize that these species of flighted birds arose from a common ancestor. Guys, when they bring a question of evolution, make sure that you relate to the question they have asked you. Don't just write. Don't just write. Don't use, now you find out that someone is using giraffes here. Why should you use giraffes when they said flighted birds? Don't use giraffes. Use what is. Ne? Make sure that you use what is given in the question. Ne? Yes. All right. Let's start. The same flighted birds are currently distributed across different continents, and then the picture. So you see it. Now, if they ask you use Lamarck theory, whenever they ask you to use Lamarck theory, you talk about 2 laws, the law of use and disuse. The number 2, the law of inheritance of acquired characteristic. Yes? So, these 2 cannot be used at the same time, concurrently, never. We'll either do the use or we'll do the disuse. So you have to pick one depending on what they have asked you. So let's look at this. So I hypothesize that species of flighted birds arose from single common ancestor, which was able to fly. It means that he was able to fly, but now they are flightless. It means that they lost that ability of flying. Therefore, was it use or did they want for disuse? Definitely it's going to be disuse. Is it clear? So now we're going to say that maybe at the beginning, all birds were able to fly, because remember when you are talking about Lamarck theory, you talk about there is no variation. All of them are the same. You understand? All the birds were able to fly. So now you talk about the disuse. They reduced how frequently they were using these birds to fly. You understand? And then the wings maybe became smaller. Is it clear? They became smaller and weaker, maybe. And then they couldn't fly. You see? They passed this allele. They passed this character, not allele, because this one doesn't talk about allele. They passed this character. Which character? Of not able to fly to the next generation. They passed this character of not be able to fly to the next generation. Then leading to these birds which are not able to fly. So that's how you're supposed to explain this. In this case, they are bringing about disuse. What if they ask you a question that, how did they, maybe they have, how did the elephant came up with the long trunks? At first, all elephants had short trunks. The elephants started to use the trunks to get water, yes, and defend themselves frequently. So you see now, in this case, they are using it. They are not misusing it. You see, frequently, the trunks became bigger. You understand? And then they passed this character of big trunks, big long trunks to the next generation. That's now the inheritance of the acquired character. Don't tell me that they passed the character to the next generation. Which character? They passed the character of big trunks. In this case, they passed the character of not able to fly to the next generation. You see now, the reason why people lose marks here is because they don't apply what is being asked. What they do, they just write. When they write, they think that they have killed the paper. And the paper has killed them. So tomorrow, you have to kill the paper. The paper can't kill you. So make sure that you apply. When you apply, you'll be able to smash the paper. Is it clear? Yes. So make sure that you guys, you apply. Why is Lamarck's theory rejected? Organisms don't evolve because they want to. Now you have to tell us. In this case, you tell us that the flightless birds did not evolve because they want to evolve. You see, you are linking the question to what you know. There is not enough evidence to show that the flightless birds acquired this character, or they pass their genes, they pass this character to the next what generation, like from being flying birds to flightless birds. There's not enough evidence to support that. There is not enough evidence to support that these flying birds, they became flightless, by not using this character. You understand? By not using this character of flying, so they became flightless, just like that. There is not enough evidence to show that. Don't just say that there's not enough evidence to support the theory. How? Which evidence? It's too broad. Ne? Yes, so it's too broad, so we won't give you a tick. But when we are writing it in the book, we say that organisms don't evolve because they want to. Yes, but now which organisms? In this case, we are talking about the flightless birds. They are talking about maybe the elephants I talked about. Elephants did not, there is not enough evidence to show that elephants acquired long trunks through using it frequently. You see? Yes. And then the elephants did not acquire the long trunks because they wanted to acquire it. You see? So you have to apply so that you are able to obtain a mark. Is it clear, guys? Distinction material, ne? Someone saying this. Sir, if use both, guys, that money is helping us to buy data, to do this, so that at least, the channel can continue. You have to support the channel. To support the channel, how will it be supported? Data, systems and stuff. Yeah. Sometimes you just do like donation. Yeah, donation. 20 rand, guys. It's not that a big deal, ne? Just donate and then get the resource, ne? And actually, you don't only receive that, you receive a lot. You ask questions, we answer questions. And guys, someone is asking me a question. Let me see if I have that question here. If I have that question, I'll try to answer it. If I don't have that question, unfortunately, but I had it. Let me see the one for flightless birds. How did they become? I think Do you mean this one? I think I don't have the question. I have to look at the question. Who is this asking me? Who is this asking me? Who is this? Who is this asking me? Olwetu. Magula. Or Mahula, I don't know. Yes, I don't have the question here. I would have explained it, ne? But guys, make sure that you apply using that, ne? Make sure that you apply. Ne? Yes. Number 2, let's go to natural selection. We go to punctuated equilibrium, ne? We go to natural selection. We go to punctuated equilibrium. We go to speciation. We go to present-day evolution, just to show you how to master it. Yes? Yes. Don't go astray. Give differences between Lamarck theory and punctuated equilibrium. Here you talk about punctuated. No. Lamarck theory and the Downing theory. Lamarck theory, organisms evolve because they want to evolve. If I want to be tall, guys, I will just stretch my head and then I become tall. Ne? Organisms evolve because they want to evolve. That's what Lamarck says. Don't say, no. You're looking for Marx. Then Darwinism say that no, organisms evolve because of natural selection. Is that clear? Then Darwinist theory, you can talk about the whole population change. Ne? While Lamarck theory, individual in a population change. Ne? Just mention 2. Go when you know 2, not more than that. Describe how Lamarck could maybe explain how did the elephants came up with the long trunk. Let me just use this example this girl is asking me. I hope I can design a small question. Please describe how the high flying small ancestral bird evolved into different species of large ancestral flying birds, flightlet birds. Is it clear? That's how you can explain. But still, you can use this and ask you describe how did they become new species. That's the challenge. You know what makes the students to lose marks? When they bring this question, they don't know whether they're going to explain speciation or they are going to explain natural selection. That is the problem. Describe how Lamarck, describe how Darwin explained this. Guys, that is the natural selection. You talk about the first point, you talk about variation. There was variation among these organisms. Which one? These birds. Those which are able to fly and those who are not able to fly. Those which are able to fly, they were being detected maybe by the predators, né? And therefore they are killed by the predators. Then those which are not able to fly, they are able to hide from predators, and then they survive. They reproduce in large numbers. This is called natural selection. Those which are not able to fly, they pass their genes of not able to fly. You tell us which gene, which character of not able to fly to the next generation. After many generations, all the birds were able to fly. That's number 1. Now, if they ask you the same question, they have brought the same question like this, and then now you don't know whether they're asking you whether you are going to explain speciation or what. Now, here they'll ask you, how did these species form? Now you are talking about speciation. Is it clear? Now that's where you talk about there was one population of flying birds. There was one population of flying birds. Yes, with a common ancestor. Yes, population of common ancestor that were able to fly. There was one population of a common ancestor or a single ancestor that were able to fly. Now we are describing speciation. Yes, the common this population was split into different continents by continental drift or by the water. Yes, by the water. Yes, mass. These populations or subpopulation or individuals of birds, they experienced different environmental conditions. Natural selection occurred independently. You understand? There was no gene flow between or among these birds. Is it clear? Therefore, they became genotypically and phenotypically different. If you remove this barrier of water and you make them mix, they are not able to interbreed to produce fertile offspring. Therefore, the ram, the ostrich, the im, and the quickie, they became new species. Do you see now how you explain? When they ask you to explain how did the species form, think about speciation. When they ask you describe using natural selection, you describe Darwinism, you explain what? You explain natural selection. Natural selection, you start with the variation. You tell us the variation between the two. While speciation, you start, there was one population. Yes, and then it split. You tell us what split them. Don't just say it split by geographical barrier. What is geographical barrier? What do you mean by geographical barrier? We need to know which geographical barrier split them. Is it clear? I think now I'm clear there, ne? Hey, someone is someone is asking me which predators can eat the flying birds. Hey, are you trying to mean that the eagle can't eat those small birds? Yes, it can eat the small birds, Baba. Yes, it can eat. There are so many. Are you trying to mean that if you are the birds which are flying up, they are not being eaten by predators? They also have predators, ne? Yes, they also have predators. They have, yes, which can, yes. Yes. Guys, let's go to the next question so that at least, I think, guys, I'm clear there. Natural selection is covered, and then speciation is also covered. Guys, don't make a mistake to describe speciation without telling us what made them separate. Don't go to the paper without knowing. Here, let me just show you here the different isolation mechanisms. Different isolation mechanism. Let's talk about this is the difference between Lamarck and the Darwinism. Yeah, the different isolation mechanism, artificial selection. Yeah. These ones. Yeah, even artificial selection. No, that's not what they want. Yes, these ones. Yeah, breeding at different time of the year. Species courtship behavior, adaptation to different pollinating agent, prevention of fertilization leading to infertility of the offspring. Don't go there without knowing these different isolation what mechanisms. So you guys, I know you're gonna make it. Eh? Don't stress. Read the question. Remember what I'm saying. Have enough time to sleep so that you can remember paper 2. I'm telling you, you come out of the paper when you are happy. When you come back tomorrow here to discuss what we have talked about, you know that yes, you will make it and you will pass it, and I'm sure you will be grateful for this lesson we have. Hi guys, let's wind up with this so that we finish up with natural selection, and I show you how to remember the names of the scientists who discovered those. Yes. Guys, present evolution can be brought into theories. Use of DDT, that's the mosquito, use of the bacteria, or use of the antiretrovirus, ARVs. But all of them, they work together. They work the same way. It's the same way, natural selection. So when you talk about this, evolution in present time, that evolution is taking place, it's talking about the resistance. So it's about the resistance. So how did the mosquitoes become resistant, or any example they have brought, and then they're talking about resistance? You talk about there is there is variation among the mosquitoes. There is variation among the bacteria. There is variation among the virus. Those which are able to survive, those which are resistant and those which are not resistant. When the application of DDT is done, yes, those which are not resistant are killed in large numbers, while those which are resistant, they survive in large numbers. Yes, those which survive, yes, they reproduce in large numbers and they increase their population of resistance. The continued use of this application you have talked about, RVs, DDT, whatever the question is asking you about, yes, will have little effect on those organisms you are talking about. In this case, they are mosquitoes. It could be bacteria, it could be virus. Hence, the resistance of mosquito has increased. So basically, that's what you talk about. So it's not that a big deal, but let's know how to apply. Variation, you tell me which variation is it? Those which are able to survive and those which are not able to survive the DDT. Those which are able to survive the antibiotics and those which are not able to survive the antibiotics. You see? Those which are able to survive the RVs and those which are not able to survive the RVs. Those which survive the RVs, they reproduce in large numbers. While those which are not able to survive, then they die in large numbers. Then those which are able to survive, reproduce in large numbers, they pass their gene of resistance to the next generation. Understand? Continued use of these ARVs or antibiotics or DDT will have no effect or little effect on these organisms. Therefore, the resistance has increased. So that's what you talk about. Let's go to yeah, guys, let's go to the next question, which is the next next part, the last part, the last part of human evolution, the last part of human evolution. In human evolution, what you need to talk about is you need to talk about the family to which humans belong. So this one is cool. The family to which humans belong. Did I say family, not species? Family, hominidae. And then the species is Homo sapiens. So this is basically what we usually ask you about the human. Writing the scientific name, capital letter Homo, and then small letter sapien. Yes. Phylogenetic tree, we'll talk about it at last. At least, guys, you know these characteristics, but don't forget that sometimes we concentrate only on the head, only the forelimb, the characteristics there. So you have to know here, you can't just say that, let me just, know, I just, two. But these are the things you have on your body. So you just think about, when they ask you about the hand, look at your hand, and then you start describing. Yes, the fingers, opposable thumb, you have the nails instead of claws. You just look at your hand and then you describe your hand. When you talk about the head, you talk about the head. Ne? Look at your head and then study large cranium or large brain, plants, eyes in front. So you look at your head and then you describe exactly what you have. Then you get the marks. Ne? It's not a big deal there. Ne? Then this one, guys, this is the difference. How does the cranium lead to intelligence? We say that large cranium means a large number of brain cells, which means that you higher intelligence. So if you have a large cranium, it means that it encloses a large number of brain cells, and the more brain cells, the larger the intelligence, or the higher the intelligence. Or more brain cells suggest a high intelligence. So basically, that's how you can talk about how is the cranium linked to intelligence. So large cranium, more brain cells, large intelligence. Difference between the skull of human and the other primates, please know them: sloping face, this flat face, the canines. These ones, guys, I'm not going to talk about too much, but there's something which I need to talk about here. Position of foramen magnum, we say that is in forward position. Foramen magnum is in forward position. We don't use any other word. Foramen magnum is in backward position. That's it. Ne? Yes. Then prognanthus, the protruding jaws. Look at the jaws here. The jaws are what? Are protruding out. If I do like the line here, you see that here the protrusion is too much, you see? So here the protruding jaws, yes, well here is just straight. Yes, so this is the protrusion of these jaws. Check this black thing, ne? It shows you exactly what happens, ne? Check this black color, ne? You'll see that, hey, it protrudes what? That color, yeah, it protrudes out. Check. Yes. And then, what is this? Broridge. This is what called the broridge. Pronounced broridge. While here, broridge is not pronounced. So you guys, there's something which I need to talk about here. Don't say that less prognanthus. You see this word? This word was no. You say that less protruding jaws, you can say less protruding jaws, but you don't say this one, this part, ne? Yeah, is out, ne? Because we updated this after the book is out, ne? Yes, so we have to update then next year. Yeah. So this is, don't say less prognanthus, we say not prognanthus for the case of human, and then here you say prognanthus, don't say more prognanthus. Or you can say less protruding jaws is correct, and then more protruding jaws is also correct. But don't say less prognanthus. You say non-prognanthus, here you say more, you say prognanthus. Foramen magnum in backward position is shape of the spine, that's all cool. Then here, walking on two legs. Walking on two legs or walking on two hind. The word hind must be there. Walking on two hind limbs. Yes. Or lower limbs. Lower limbs. You can say lower limbs or you can talk about hind limbs. So make sure that when you are defining what is bipedalism, so that is also a small update so that you don't mess it up. Factors that affect bipedalism, we say that the first one is, please know them. Position of foramen magnum, the shape of the pelvic girdle, and then the shape of the spine. S shape allow flexibility. I explained this very well. Shape is short and wider to support the upper weight. The position of foramen magnum is in forward position to allow the spine connect vertically. Yes, I'm not reading here. You might be thinking, but still when you read, not supposed to be like cramming. You just need to know. You just need to what? To know. Sir, this lesson is recorded. They're saying, can you explain why most predators are less likely to feed on? Now, where is that question? Guys, don't ask questions which is in the passage. I don't have the paper. Unless you're asking a question which is an open question, yes. But now if you ask me a question which is in the passage, I don't know the question you are asking me. So is this lesson recorded? Yes. It will be, you can only play it offline, maybe after a certain time. Or I will do a copy of this so that the copy can be downloaded offline. Yes, so that you can watch it offline. But you have to come back maybe after like an hour. Yeah, maybe around 7 o'clock, then you will be able to play it or to download it. Yes. All right. Let's continue to the next question. Yes, so you guys make sure that you know. Then advantages of bipedalism. Difference between bipedalism and quadrupedalism. This one is obvious also. For position of foramen magnum in forward position, this one is in backward position. This one is S shape, this one is C shape. Short and wider, long and narrow. So you guys, you have to know that. I want to talk about the advantages. At least go when you know 3 of them. Free hands so that you can pick food, the standing upright, the eyes are high, the ground, so it's better view. You understand? You see, you think why ladies like to put on high heels so they can see far, man. Can see far. They want to see far. Why? Because you're off the ground, so that you can see exactly what is happening there. I don't know what you're looking for, but those who put on the high heels, you must tell us what you're looking for. Anyway, paper 1 is done. There's no reproduction here, so I won't talk about it. Yes, but anyway, yeah, bipedalism so that you show us. They are trying to show us. Have you ever seen? Let me leave it. Because people that will say that, you guy, this guy is doing this. Okay, let me leave it. But basically, when you walk, you show us, you display the 6 organs so that, you see that, this is my feet or this is not my feet, something like that. Yes. Yeah, some questions. Disability, observe a difference between scale 1 and 2. Guys, I think you talk about this, you talk about the canine. Guys, if they bring it like this, don't talk about the position of foramen magnum. Do you see any foramen magnum here? No. You only talk about foramen magnum if you see that, yes, I can see. The characteristics of the upper limb, you see what I talked about? They're talking about the upper limb. Explain how increase the brain volume lead to intelligence. I talked about that. That increase in the cranium increase means that more brain cells, which more brain cell leads to a high intelligence. So what is bipedalism? Walking on two hind limbs or lower limbs. Explaining how each of the following skeletal have continued bipedalism. Is position of foramen magnum is in forward position to allow the spine connect vertically. Pelvic girdle is short and wider to support the upper weight. S shape to allow flexibility. Yes, allow the flexibility of the organism. You see, so it's a simple thing. Lastly, but not the least, the evidence of hominids. Fossil evidence, genetic evidence, and cultural evidence. Guys, there is a difference between don't say when they ask you about the evidences, you don't say the mitochondrial DNA. You don't talk about tools. The evidence is genetic evidence. Example of genetic evidence is mitochondrial DNA. The evidence is cultural evidence. Example of cultural evidence is use of tools. Is it clear? Yes. So fossils remain fossils. Yes. Don't talk about Australopithecus. Those are examples. This one, it kills people. Kills people, a transitional fossil. What is a transitional fossil? A transitional fossil is that fossil which have both characteristics of the present species and the species before it. Don't say that it has the characteristic of present species. No. It has the characteristics of present species and the species before it. You understand? Yes. So you must link the two. How? Or they can bring two skulls and they ask you, give a reason why this is classified as a transitional fossil. Let me see if I can find a skull here, and then I show you what I'm trying to say. You think there are no skulls here? Explain how colonial drift relate to common ancestors, our speciation lead to photos, lemurs become different species. So it's the same thing which we was trying to explain. Yes. So you see now, here you can talk about the position of foramen magnum because now you can see. Here you can talk about the C shape. You can talk about the rectangular shape of the jaws. Here you can talk about the position of foramen magnum. You can see it. Here you can talk about flat face. Here you can talk about the sloping face. Talk about the flat face because you talk about what you see. Yes. Here you talk about the jaws. You see exactly that you can see them. What is a transitional fossil? You see. Now, what shows that this is a transitional fossil? You can talk about, maybe they can say that this is X, this is Y, and this is Z. Z, Y is a transitional fossil. Give a reason for your answer. Because it has both characteristics of this and this. So, but now you can talk about that because now they have given you a scenario what to use. Pick up the character. Look at the canine. The canine of Z is not as big as this, but is not as small as Z. The canine of Y is not as big as X and is not as small as Z, meaning that it's just there in between. You see? Yes. The shape, this one, is not as rectangular as X and is not as C-shaped as Z. You see? Yes. So basically, you have to link the two. When they ask you, please know how to apply. Don't just use what you know from the head. Use what you see in the diagram. Is it clear? Yes. Let's go to the fossils and then we finish them. How do I remember this? This is the table where you're supposed to remember. This is the table where you're supposed to remember. Adipithecus, the first one and the last one is discovered by the same person. Adipithecus, Ramidus, Homo sapien. There is a small video I created which shows this. It's just like it's like a song, yeah, but it's you are remembering, yes. So so that is the first step. So basically, Adipithecus is Tim White in Ethiopia, and then Lucy, who discovered Lucy? Now, Lucy is Johansson, is in Ethiopia. You understand? Johansson, Ethiopia. Actually, the first 1, 2, where were they discovered? In the country where there was no colonization, a country where the one which was not colonized. You think about that country, and it's Ethiopia. Yes. Number 2, now you have 3 Australopithecus africanus species. All of them, who discovered them, they start with the R. Raymond Dart, Robert Broom, and Ron Clark. All of them are starting with R. Is it clear? So they cannot ask you home chart, rate of food, Mrs. Place, and then you start thinking about Lady Bagger. All of them are starting with R. Is it clear? Yes. Taum child, Raymond Dart. It has the A, it has the A. Is it clear? Yes. So now you know it. So now the rest you can know. Ron Clark, little foot. And then Mrs. Place, actually it's called Mr. Place, but I don't know if they credit if you say Mr. Place. But the recent one is supposed to be Mr. Place. But because the books, but when you are trying to do the discussion, they credited it that is Mr. Place, but it is Mrs. Place until we are told to remove it, to update it. But we will not update it until they tell us to do so. So when you say Mrs. Place, it's cool. When you say Mr. Place, I think it's also cool because lastly, they credited it. Yes. So think about it. Then Australopithecus sediba. Sediba is Karabo. That one is Lady Bagger. Please, you need to know that. Homo sapien had a man, and then Homo erectus. Please watch that small video. We will be able to know. Yes. But now, how do we use this as genetic evidence? These 6 marks, these 5 marks, please. Evidence to support the out of Africa hypothesis. How do I use this? Let's go to the next question so that we wind up. And they're saying that fossil, guys, you need to know this. Let me just go here straight. If they ask you to explain the out of Africa hypothesis, guys, you need to know that this is modern humans originated from Africa and migrated to the rest of the world. That is the first point. You need to know that. But now, when they talk about evidence to support that, is fossil evidence, genetic evidence, and cultural evidence. Fossil evidence, you talk about the oldest fossil, Adipithecus. Ne? The Adipithecus, Australopithecus, and the Homo habilis. All of them, they were discovered in Africa only. Adipithecus, Australopithecus, and Homo habilis were only discovered in Africa. But now when you talk about oldest fossil of Homo erectus and Homo sapiens were discovered in Africa. The oldest Homo erectus and Homo sapiens, you have to add that word oldest. Is what I say. Also, fossils of Adipithecus were only found in Africa. Fossils of Australopithecus, Rabo, little foot, child, you can talk about too. And the Homo habilis were only found. You see, this word only is very important. But once you come to Homo erectus, you talk about the word oldest. The oldest fossil of Homo erectus and Homo sapiens were found in Africa. So these two, we want the word oldest. If you don't put it, you cross it. And then this one, you don't add the word oldest because we will ask you what about the newest? So we cross it. So don't add here the word oldest. Guys, here we are too strict. Here, you only talk about Adipithecus, Australopithecus, and Homo habilis only found in Africa. But when you talk about Homo erectus and Homo sapien, you talk about the oldest fossil of Homo erectus and Homo sapiens we are found in Africa. Mitochondrial DNA, that's genetic evidence, mitochondrial DNA. Now here you just talk about where the oldest female was discovered. Is it clear? Yes. Then cultural evidence, you talk about the oldest tool, which is Odo 1, were discovered in Africa. Yes. So basically that's what you need to talk about. Yes. Lastly is the phylogenetic tree. I'm going to show you the questions in this booklet. The questions they can ask you, they won't ask you apart from these questions given you. This book was designed to make everyone pass, and not just a pass, but you pass with a distinction. Eh? Yes, you pass with a distinction. Lastly, you guys, are you listening or you are busy asking? You are busy asking numbers? Muzanzi. All right, guys, make sure that you make Muzanzi shine. Eh? Yeah. All right. Here are the questions. These are the questions we're going to talk about. What is the name of the diagram given above? It's called the phylogenetic tree. How many genera are shown? When they talk about genera, you're looking at is one. Adipithecus is the first genera, Australopithecus the second. The first name, Australopithecus, second is one. This is two. Then 3, Paranthropus. Do you have Homo? 4. Do you have another one? Homo? I think there are 4. Unless there is something I've not seen, but you look at the first name and you don't repeat it. So we are done with the genera. What is the common ancestor of Homo sapien and Homo erectus? Common ancestor of Homo sapien, Homo erectus, and Homo sapien. So you move back until where they join, Homo ergaster. So Homo ergaster is the common ancestor of Homo sapien, Homo sapiens, and Homo sapiens and Homo erectus. Then the next one is, what is the common ancestor of all species? Here you just look at it. Is the common ancestor is Adipithecus remidus. It's the common ancestor of all species. Eh? So it is easy that way. Then the next one is they're asking you, give what is the common? Give 2 pairs of species that competed for resources. When they say 2 pairs of species that competed for resources, they're talking about 2 species which lived at the same time. Eh? Ready? Yeah. So for example, you just choose. If I draw a line here, I will see that this species, they existed at the same time. So you just look at the species which existed at the same time. Eh? Yes. Then they're asking you, so then name 2 species that competed. Okay. Which species first used tools? Guys, this question depends on the question they have asked you. The species which first use tools is Homo habilis. But if they give you a diagram and then they show you that, okay, this is the other one tools. These are the species, okay, this other one tools. If this is, where's the tool? The tools are here. So it's 2.5 billion years. Yeah? This is 2.5 billion years ago. So which species first used tools? I have to draw a line and then I find out which species first used tools. So here is showing you is Paranthropus ethiopicus and also Australopithecus afarensis. Afarensis africanus, something like that, based on the diagram. You understand? Yes, but if there is no diagram, then it is Homo habilis, the handyman. Then they are asking another question. Species to walk on 2 limbs. The species to walk on 2 limbs is Homo erectus. But if they give you a diagram, you have to look at it. Bipedalism is here. So which species first walked on 2 limbs is the Australopithecus anamensis. So Australopithecus anamensis is the one which first used the 2 limbs. So Then they're asking you another question. How many million years ago did Australopithecus africanus appeared? The Australopithecus africanus appeared. Yeah, when you talk about Australopithecus africanus, Australopithecus africanus, they're saying that which species first walked? Okay, how many million years ago did Australopithecus africanus appear? So you come back here on the diagram and then you see where did it get appeared. Australopithecus africanus, where is it? Africanus is here. Ne? When did it, which one? Yeah, it's here. So you find where did it get appeared? Maybe is 3.2. 3.2. Remember the 0 is this side. So you count going downwards. Ne? You count from 0. 0 point, maybe is 3.2. Yes. Million years ago. Million years ago. Then they are saying that when did Homo habilis became extinct? Now you look at where it got extinct. Homo habilis. Where is Homo habilis? Homo habilis is here. When did it get extinct? So you draw a line touching here, and then is maybe is 1.6, because 1.5 is around there. Maybe it's 1.6 there. You understand? That's when it got extinct. Then they're saying that how many million years did the Australopithecus africanus existed? Here now we don't say million years ago. You just tell us how many million years, because it's like asking you how old are you? You're telling me 20 years ago. No. Australopithecus africanus is here. So now I have to come where it started, maybe 3.2, and where it ended, maybe 2.5. So I'll say 3.2 minus 2.5. Maybe it's going to become 0.7 million years. Not million years ago, because they're talking about how long did it exist. Yes. Then they're saying that how does the brain size show evolution trend? We talked about that the brain size means that the higher the brain size, the higher the number of cells which lead to more intelligence. Give one example of transitional fossil. An example of transitional fossil is Carabo. And talk about Carabo, Australopithecus sediba. How does dentition show the diary of the organism? Dentition shows the diary. How does it show? If you have sharp canines, it means that you are feeding on raw or raw food. Yeah, food which is raw. Understand? Yes, raw food. And this food is tough, therefore you have to use sharp canines like this. Sharp canines, yes. I think that's the question. Then let me just go here. Just lastly, how may name the family to which the organisms belong? Hominidae, and then describe the cultural evidence. We talked about it. How long did it, most recent, how long ago? So you talk about how many years ago. Yes. And then explain the possible reason why Homo ergaster was placed between Australopithecus afarensis and Homo. Then it means that it's a transitional fossil, therefore it has characteristics of both this and this. Yes. Explain why fossil of organisms that existed from 4 million years ago to present time are used to support out of Africa hypothesis. How do they explain how? So from 4 million years ago, therefore you talk about the oldest fossil of, you talk about the fossil of Australopithecus afarensis, Australopithecus, just say Australopithecus, and the Homo habilis were only found in Africa, while the fossils of Homo sapiens and the Homo erectus, the oldest, were found in Africa. Basically, that's what you need to talk about. Guys, I think I'm going to stop here. I wish you the best. I wish you the best. I wish you, I repeat, I wish you the best, and I bless you with the distinctions. I know that you'll make it. Don't let me down. Remember what I've said. Watch this video again, so that, because when you watch it, basically you are revising. You understand? You are revising, and I know that you're going to make it. May God bless you, bless your hands, and bless your future. I stay as Saidi. See you again at the university courses. And tell your brothers and sisters to join the YouTube so that those guys can be helped the same way you are helped. Thank you very much. Until we meet again.